Okay, yeah, so let's get started again. So we talked about the sort of spontaneous strains and sort of potential generation of shape and mechanics through spontaneous strains in the first hour. And I gave you this sort of quasi 1D example of a model that sort of captured really the relevant sort of mechanics and biology of this formation cephalic furrow and fly. Hopefully it's motivation to sort of convince you that like this sort of idea of changing lengths in the tissue is a good thing to pursue. Of course, there's no interesting network there yet. I mean, technically it is a network, but a very boring one. It's a chain network model. And then I introduced on the blackboard the sort of key elements and properties of the vertex model, which is like one of the major standard theoretical models that people use for epithelial tissues, especially for the apical junctional network of epithelial tissues. Okay, and so now we're going to move on to talking a little bit more about how some of these sort of active stresses and actively mediated spontaneous strains can occur and sort of interact with a vertex model setting. So we're going to stay in the sort of purely theoretical realm of these vertex models for a little while. And then we'll close with a more complicated, another full biology story with a more complicated network than this first case, where we have a real, epithelial apical network doing active behaviors that drive a shape, and we'll try to understand it. Okay, so let's move ahead. Okay, so this is a busy slide. So now this on top here, right? So this is just a reproduction of the vertex model energy, right? That's I already wrote down. And so, what was going on, there's some work I did together with Frank Ulicker and some of his postdocs, and also Namdar from India a few years ago, thinking about like, all right, if you have a vertex model, and so here we have a vertex model with periodic boundary conditions in both cases, how within the model can you drive these topological network events, right? So obviously you have to come up with, as I said before, you have to come up with some kind of rule, right, to do it. And so what we had sort of in this work, we sort of thought about two different ways we could couple the elements of the vertex model to something anisotropic to drive directed versions of these exchanges. Okay, and so on the left here, right, we have anisotropic bond tensions, and so this is the bond tensions, are implemented in such a way that they're not, just constants or just sort of simple values, but they come from some polarity vector that couples to some external field, right? So we have this colon here is the sort of, it's the complete collapse, right? It's the trace of the multiplication of the tensors, right? So you get a scalar out of it. So you totally contract everything. Yeah, so you couple some kind of in-plane polarity, right? Or in this case, the direction of the bond with some external field, right? So that the bond tensions then become greater, right? If they're aligned one way with respect to the field, and smaller if they're aligned some other way with respect to the field. Okay, and if you do this, right, you look, you implement some time scale into the system, right? So you have some dynamical simulation going on, and you look at the different sort of components of the energy and components of the stress on the sort of periodic box, and you see that you get a bunch of it coming from cell rearrangement, red is cell rearrangements. So you get these sort of directed, anisotropic stresses coming from the rearrangements. And at the same time, you get this sort of counter response in the cell elongations, right? So the cells rearrange, kind of, up to down, but then they also elongate at the same time. Oops, in a countervailing direction. So this is the fixed box boundary conditions. So the box you have a periodic box and you can't change its dimensions, which means that of course you have to get some back reaction. Right? Everything has to balance. And the total shear strain or shear stress has to add up to 0 because the box itself doesn't shear, right? Like you don't get any shape change in the box. So, yeah, you get essentially that the T1s and the shape changes sort of counterpose each other, okay? Now, and then you can also do it, you can think about driving this with anisotropic cell stresses. And so here, right now, you have, again, some external field that you couple to, but you do it with the shape tensor of the cell, right? So the cell itself can be elongated in a certain way or not. And you sort of drive elongations of the cell along some external field, okay? Or some external vector, anyway. And you get a similar thing, although now you drive, you know, the positive, the forward direction, the backward direction is swapped, but you get cell elongations and neighbor flips to maintain the fixed box boundary conditions, okay? So you can sort of couple, and again, now in both of these cases, I should say that the actual topology change is still being put in by hand in some sense, right? Like the topology changes occur, you know, in both of these scenarios once individual edges are driven to be so small that one then does the Monte Carlo check and see, okay, do I rewire the edge? Do I change the neighbors or not? And then you take the, if the energy is the change is good, then you know you take it, and if it's bad, you still take it with some multiple factor, right? And then you continue evolving. All right, so the point is that like you can think about, there's ways to think about coupling the mechanics to some external signals to drive in a vertex model where you can drive these sort of events. But this is still just sort of a monolith, right? We're not thinking about shape outcomes here yet. And so in order to get shape outcomes, what you actually need, right, is for, you know, different locations to create, you want to be able to create these anisotropic patches, like in what I showed you before, right? You had like an isotropic piece of like the pneumatic elastomer, right, with an elongate, you know, there's a longer one way and shorter another way, for example. And so, and I had this sort of leading analogy to convergent extension where it's known that if you get a lot of these cell rearrangement events going in the same direction, you elongate the tissue one way and you draw it in the other way. But people have not ever really looked in the vertex model sense, you know, at a sort of a tissue patch with open boundaries, like what is the functional relationship between the sort of rate of directed T1s versus the shape, you know, the local shape change that you get. And if one can sort of coherently construct that map, then you can think about putting together patterns, right? If you have the T1s going, instead of all going the same way, if they're going radially around a topological defect, for example, then, you know, you can mediate some kind of shape change. And so we did two kind of different approaches here. This is with postdoc Carlos Duque, where we started with a sort of vaguely, you know, an isotropic tissue patch, like a circular tissue patch, and we drove a bunch of active directed T1 events. And I'll give you the sort of distinction on the right in a second. And at each step, we allowed them to relax, right? So we had an active T1 event, and then we do the relaxation, you know, from the energy from the vertex model. Oops. And then we go again, then we go again, then we go again. And then just to sort of, you know, double check, you know, in response to reviewers, that you could sort of build up, you know, kind of save up the desire to kind of flip all at once, and then flip sort of all at once, and then relax the whole thing. And that you would still get, you know, a sort of a slightly modified version of this anisotropic change, but you would still get some anisotropic change. And so, yeah, so there's, in the end, what we realized we needed to do was consider three different types or classes of these neighbor rearrangements. So there's active directed, right? So that's clear. We have some predefined direction, you know, where we want them to be more likely to have a rearrangement, you know, with the edges aligned with that direction. We want to push it in that direction. And so, you know, so for this, we don't worry so much about the intermediate mechanics driving, you know, the fact that you can drive these things in a more continuous way. We just worry about, okay, we know that we can do it, so now we're just going to do it essentially by hand because we're worried about the collective effect. So we have a probability distribution that's sensitive to the orientation of the angle, and then we randomly choose, you know, in a sort of in a stochastic way. You choose an edge, it's going to be the next edge to flip, and it's much more likely for these directed ones. It's very much more likely to be one that's aligned a certain way versus the other way. Okay. On the second class is active undirected. It really just literally is we randomly choose an edge, any edge, and we flip it. We do the T1 flip. Right. So this neighbor rearrangement is equivalent to what I keep saying, flipping the edge, because this edge is horizontal and then it flips to vertical, right? You rewire it by flipping it to vertical. That's why I'm saying flip. So active undirected is really just a completely random T1 in any location, right? Regardless of orientation, just totally flat background. And then the passive ones, right, are, in both of these cases, we do the neighbor rearrangement and then we allow the system to relax, right? And during the elastic relaxation of the system, it is possible, right, that one of the edges might be driven to become very small. And if that's the case, then we allow it to initiate a neighbor change in the same way that we talked about before, right? Now, we work primarily deep in the solid regime of the vertex model. So if you're close to the solid fluid boundary in the vertex model, then sometimes these things avalanche, right? So you get one rearrangement and that rearrangement causes another, and causes another, causes another, causes another during the relaxation, right? But that's a property of being sort of either in the fluid regime or very close to the fluid transition. So we're deep in the solid regime. So for our simulations, we do get passive rearrangements, but they never, essentially, I don't think Carlos ever saw an avalanche. We saw, every once in a while when we drove an active one, we would get one passive one in response. Most of the time we drove an active one, there was no passive T1 in response. But you do get them sometimes, just for completeness. Okay, so now, why do we even care about this undirected one? Isn't the whole point to drive a shape, right, with these directed T1s? Well, indeed, you can drive a shape with the directed T1s, right? So you can take, this is a plot of the number of T1s on the x-axis, sort of normalized by the square root of the number of cells, and the strain, right, the effective spontaneous strain that you generate on the y-axis. And so, and then the different colors are for different sizes of patches, right? And so what we get is that, like, if you just do more and more and more directed T1s, right, you drive out the patch until, you really extrude it out as far as it can go, right? The fact that it turns over here, these turnovers, these are finite size scaling effects that come from the fact that eventually the tissue gets so extruded that you can't make it any more elongated, right? Like if you have a tissue patch with 100 tissues, 100 cells in it, by the time you're, 2 by 50 or 3 by 30, right? Like there aren't any more available T1s to make it go any farther. You can only go so far because you have individual units making up the system, right? And so, and very nicely, all the different size systems, you have this nice curve collapse, and you have this similar behavior, all sorts of Poisson ratios. There's this well-described, functional relationship that occurs between the driving and the formation of a spontaneous strain in the system. But it would be interesting to think, like, okay, you hit some limit, the number of T1s to hit the limit is pretty big, but, there are lots of tissues where you see larger numbers of T1s. And also, if you want to have a specific magnitude of a spontaneous strain, right? If you're making a shape, right, and you actually care about the details of the shape, then you need like quite fine control, like good fidelity of this number of T1s that you're performing to get the right spontaneous strain, right? And so there are ways that biology could be counting, right? Maybe there's some rate and there's also a clock, right? For example, so you have rate over time that you count, it's simple. But we thought like maybe there's some way to get a steady state instead, right? With different steady states landing on different spontaneous strains. And so that's where we hit on the idea of mixing these sort of directed and undirected rearrangements, right? And so just ignore the plots on the right for a moment, right? So if we have some state, right, some positions in the system, right, and we apply like a whole bunch of undirected rearrangements, over and over and over again to our original state, right, in the reference space, the reference space target space. then this is equivalent to, adding up a whole bunch of strains, right? So you have a bunch of small random strains that you add up over and over and over and over again. And of course, so long as those random strains are independent and identically distributed, these are the ingredients for essential limit theorem, right? And so in essence, this is the same as the physics of a polymer, right? Of a random string model, except that it's for a tensor instead of a vector field. And so this would predict, right, that the strain of these systems should look like a Gaussian, right, for the density of states, right, of these systems should look like a Gaussian for all these different drivings if we include the undirected active rearrangement. So we're driving a directed rearrangement, and we have some active background that acts like an effective temperature, right, that lets you mix up ergodically mix up the possible states. And you fall into, you're going to naturally fall into the thickest part of the density of states because that's just where more stuff is, right? And indeed, that's what you see. You look at this G of E as the density of states, and these are, P of D is the ratio of directed to undirected active transitions. So as you get more active, you generate more strain, but you always have this nice Gaussian profile of, and this is the absolute value of the strains, that's why the 0 ones pile up. This beautiful Gaussian profile of the density of states for the strain, right? So it's an entropic tissue is what we call it. And so now you can do these simulations many times for many tissue sizes and many different ratios of the directed and undirected active T1s, and generate these sort of functional relationships between the strain and the number of cells or the ratio. Sorry, this is the strain as a function of the number of cells in the patch, so the size of the patch, and then the colors are the different ratios of directed versus of the driving versus the effective temperature. And then this is the same information presented the other way around. So now here's the strain again, and now it's the ratio on the y-axis, and the color is the size of the system. Right, so depending on how you prefer to interpret it, you can look at either one. And then down here, we extracted this sort of the exponent, right, on the strain goes like n, the number of cells to some exponent. We extracted the exponent behavior as a function of the ratio, or we fit instead here we have some nice exponential fits, and we fit the exponential prefactor, like the rate here. And we did the same thing for the Poisson ratio, but that's less important. That's one of the parameters of the shape programming, but like the strain amount is the one that's sort of more obviously interesting. But the Poisson ratio matters too. You can also find functional forms for that. Okay, so this is all to say that, like, from a theoretical point of view, right, the ingredients exist for there to be a coherent coarse graining over either just raw numbers of directed network topology changes, or steady states and the rates of these topology changes subject to this balance between like the ergodicity coming from the completely random ones and the directedness coming from directed ones. And so now we can move ahead to a real system and try to do an analysis. Now we're not going to be able to do, you'll see why, but we're not going to be able to do quite as deep an analysis in terms of like these individual T1s or individual rates of T1s or identifying. Of course, we don't know. We just see T1s, right? In a real system, we don't know if it was supposedly random or not random or what have you. So we can't apply exactly the same set of formulas and functional relationships, but we at least know that there's a good theoretical basis to expect that they exist, to expect that you have a coarse graining that exists over the network states onto something that's more like a spontaneous strain tensor field, a continuous field. Okay, so now what's the biology? Like, what's the biology example? So we're going to focus on something called the wing disk eversion. Does anybody know what that is? Have you heard of that before? No. So the Drosophila wing disk is the precursor. It's like the limb bud for the wing. It's the precursor tissue to the Drosophila wing. The adult, mature Drosophila wing is a bilayer of cells. So it's like one epithelial sheet, and then another epithelial sheet, like right underneath it. With the apical sides out on, both sheets, okay? But at early times, in early larval growth, the sort of blue tinted tissue, that's the tissue that's going to become the wing, and the rest of this white tissue, which is much larger in early times, is the part that sort of becomes the hinge, which is like, it's effectively the shoulder. And this blue tissue is a classic single epithelia, so it's one layer, right? And at mature times, it needs to get to being flat too. It's like, well, flat one layer, but it needs to be flat two layers. And so how does it do that? It does that by making this eversion transformation. So it's a flat single layer. It very slowly develops sort of a light dome-like curvature. This is pre-eversion. So it becomes flat, and then it's lightly domed. And then eversion happens, and over the course of 4 to 6 hours, which is again quite fast on the developmental time scale, it goes from this light dome all the way out to like this, except that it's an anisotropic shape change. So it's not a pure fold. It's a smooth curvature this way, and then smooth curvature the other way, but more shallow, right? So it develops a kind of ridge line, right? And what was this line running down the middle of the disc ends up being sitting on the ridge. And then after this fast change, like this, then it slowly kind of rolls out, and then they come together, and that's how you get the bilayer. This line is important. It's called the DV boundary between the dorsal and ventral sides of the tissue. And the cells along that line are genetically distinct. They have a different gene expression program going on. And the sort of class of activities that they're performing seems to be a little bit different. We'll talk about that a little bit later. So that's the version. So Yana from the Suzanne Eaton's lab, and then later, after Suzanne tragically passed, from Natalie Dye, who took over Natalie Dye's lab. It was the main experimental collaborator on this, and she took these amazing 3D images. So this corresponds to this stage. So you see, this is the kind of light dome, and then that little light dome buds way out. Like it almost looks like a thumb sticking out now. So this is a big, very anisotropic shape change effect. You can't get this without changing the metric, right? The metric is very different. So there's no way this is any kind of fold. There's no way, in the field, it's long been known that apical basal, ratio changes aren't driving this. You can screw up like that sort of mechanism. It doesn't change anything. There's not really any important external tissues to speak of that can be doing this with external forces or instability, right? So this is a prime example of something that maybe is a good target to think about for these sort of in-plane effects. For completeness, I should say there is a very, very thin, oh shoot, I forget what it's called now. There's a very thin tissue that sort of wraps around it that it breaks through when it's doing eversion. So there is an external tissue present, but both Yana and previous people knew that if they cut that tissue away prior to reversion, then eversion it proceeds in exactly the same way. So it's also not really that doing it.
Carl.
Okay.
Carl.
So the idea then is to think about
Carl.
having some coarse-grained model of these network changes, mechanical network changes, to drive a shape. And so we have some analytic theory to sort of give some certain kinds of metric changes, but at some point it's good to have a numerical model where you can really do more crazy things. And so Abhijit put together this really nice sort of spring mesh, dashpot mesh lattice model, where we can change the natural rest lengths of the springs in some pattern, right, to capture the spontaneous strains in some way.
Carl.
And then look at the sort of force balance and, with the dashpots, the dynamics of the system to see what kinds of shapes you land on. And then for these changes in rest length from the springs, right, there are different kinds of cell behaviors can be implemented in different ways, right? So if you have the areas of the cells changing locally, right, then you can, all the springs in that area will all want to get longer, for example. Or if you have anisotropic changes like these neighborhood rearrangements, or the cell shapes themselves elongating, then in that region of the model, then the springs that are aligned with the principal direction of the anisotropic shape change will have their lengths changed one way, and the springs that are perpendicular to that direction will have their lengths changed in another way, right? So, straightforward.
Carl.
And then we can just put in some sort of trial patterns by hand and show that the model does indeed reproduce, quite good, robust family of shapes just from some simple patterns that one can just kind of think up out of the blue, right? So here's some area gradients going, high to low from the inside to the out, and here's area gradients going low to high from the inside to the out, and you get either a spherical cap or like a saddle shape for those two, or here's the uniaxial, case where you're sort of having everybody extending radially and shrinking azimuthally or vice versa, and you can get again different strong shape changes. Starting again, in order to mimic the wing disk, we started from this is the starting state where we have a lightly domed thick tissue.
Carl.
Okay, questions so far?
Yeah, Carl, it was me.
Okay,
can you hear me, Carl?
The next step, right, is much like in the cephalic furrow model, right? We had one fitting parameter, and so we want to get as much as we can from data, or make as many informed sort of specific choices in advance, like about the symmetries and things like this, to reduce the the sort of fitting degrees of freedom we have in the model at the end, so that we can be sort of more and more confident that what we're seeing makes sense if what we see matches reality, right? So, okay, one of the assumptions is that the pattern of behaviors in the tissue is going to be something radially symmetric with a topological defect, together with this special band of different behaviors occurring down the middle, which corresponds to the DB boundary, where it's known that those cells have a different gene expression program and are observed to do different things, right? So they, this is consistent with observations that the pattern kind of on average looks radial on most of the tissue and then just sort of along this strip in the DB boundary, okay? And so we assume that symmetry is exact. We assume that the initial condition is a perfect spherical cap, right? So that the real initial condition is it's not perfectly spherical, of course, it has some variance, and it's like a little bit longer one way than the other way, and there's a little bit of bumpiness. But we assume that away, we have a perfect spherical cap. And then if we make those assumptions, then actually much like in the cephalic furrow case, together with all the other actual things that we can measure, the only fitting parameter left is, again, the effective the ratio of the bending rigidity to the stretching rigidity. It's the same one. It's the same bugaboo. But one can't ever measure that, unfortunately. But fortunately, this is one, right? So that's good. Okay. Now, in order to extract the cell behavior data that we need, we have to go back to thinking of the cell apical system again as a network, and then we can make a topological coordinate system. Right, and so this actually turned out to be really critical to being able to infer the rearrangements. So the idea is that you choose a cell to be the center of your coordinate system, and we have a good way to make that choice, which we can talk about later if you're interested. But you choose a cell to be the center of the coordinate system, and then you can just look at the rings of cells. You have all the first neighbors, and then all the second neighbors, and then all the third neighbors, and then all the fourth neighbors. Right, and those generate essentially rings going out, so long as the cells are approximately the same size, and it gives you an effective coordinate system that's very much like a circular coordinate system, but that you don't need to know anything about the geometry. Right, you don't need to sort of follow the curves of the surface. As the surface geometry changes, you don't need to sort of mess with different crazy coordinate systems. It's a purely topological coordinate system based on cell neighborhoods. And fortunately, when you have a rearrangement, right, you push cells in and out of neighborhood bands, right? So you can infer, I think I have that slide suppressed, but you can infer the amount of cell, the net amount of cell rearrangements along the pattern that you're looking at with this topological coordinate system in a very direct way, right? It's perfect for that. Okay, and so we have Now, snapshots, right? So the data that we're able to take, the super high fidelity 3D data with light sheets, we can't do live imaging on, right? So we have snapshots every 2 hours, right? And so we have the sort of late larval stage, 0 hours after puparium formation, that's this HAPF business. So it's sort of larva, it makes the puparium, and then 2 and 4 hours, 4 hours after puparium formation, that's when emergence is over, okay? So this is all told somewhere between 4 and 6 hours, right? It's 0 to 4 after the puparium forms, and like from this larval upcrawling stage to the formation to the puparium, it's varies a little bit, but it's 0 to 2 again. Florian, looks like you have a question. Okay, so this is the data on top. And we evaluate, just for simplicity, we have this complicated 3D shape, so you have to do some kind of like sort of lower dimensional analysis to just make things more interpretable. So what we chose to do is look at the 2 primary cross sections with respect to that special band, the DV boundary. So we have a cross section sort of along the DV boundary and a cross section, and a cross section perpendicular through the middle of the DV boundary. And then we look at the change in curvature from the first time point, the black one, the larval stage. And then the change in curvature as a function of arc length, from the tip to each side. And so it sort of develops this very, very sharp curvature in the middle, and it gets more weakly curved away on the very ends of the region that we're looking at. And then along the DV boundary, very, very little happens. So in the middle, sort of the curvature goes up a little bit, but not much. And so there's not much happening. So that's what happens to the shape in the real system. And now at the same time, we can look at the cumulative strain pattern, like the inferred spontaneous strains coming from the actual observed cell behavior. So we can see, at each time point, we see the cells in different regions change their areas, change their shapes, and we can use that topological coordinate system to see, how much of a neighborhood flow has occurred, a net neighborhood flow. And this is why we can't use the full machinery, the theoretical machinery I showed before, because we can only measure the net neighborhood flow, right? So we don't know, for example, how much, if there's like the undirected active T1s going on, they would average out to 0 net flow because they're going every which way, right? So we have no idea, like because we're only looking at snapshots, whether or not that's going on or not. We just know what the net effect is. Okay, so it's kind of a little bit more like just the counting version, the easy version from the previous theoretical part. Okay, and so that gives you these 3 patterns with these 3 magnitudes. So the cell rearrangements look like they're pretty strong. The elongations look present, but a little bit weak. And the cell area change looks pretty strong, but it looks like from the gradient point of view, it looks weak, right? So the cell, like the overall cells all get bigger, but there doesn't look to be a particularly strong gradient in the changes of the areas. Okay, and now, a lot of other work in tissue mechanics has pointed, over and over again to being cell rearrangements as being, a good way to drive active stresses into the system. And so our sort of first thing we looked at was to take just the rearrangements and assume that they're active and put that pattern into our model, right? So we take the observed cell rearrangement pattern, and we implement that into the model. And so we drive a 3D shape change in the model. And then when we sort of do the same quantification as we did for the data, we see qualitatively we're doing pretty good, right? It's not great, it's not a perfect match, but it's a good start, right? So we have this big upwelling in the middle. It doesn't quite go down as strongly on the outsides. For the across DBB section, that's this cross section that you see on the left. The real divergence is that we don't generate as much asymmetry as there really is in the real system. We're still getting quite a bit of curvature along the DB boundary as well, okay? And so what can we do next? Well, we can think about how much, resulted with input spontaneous strains were and how much actual strain came out, right? So we were inputting the black curves, and the yellow and the purple are how much you actually get in the model, right? And so what that means, of course, is that there's some residual, right? We're not hitting exactly the metric that we're prescribing, where there's some residual amount of strain that's left over. And now look at this mapping of the residual strain. This looks just like that, right? Same color, same directions, right? And so this is a sign that perhaps, the rearrangements really are an active driver, and the elongation component is passive, right? Because the residual strains and the associated stresses that came from just putting in the rearrangements matches what we saw in the elongations. And so if it's true that the elongations are really just passive, then we would expect if we only put in the elongations, them to do the wrong thing on the shape, and also to leave almost no residuals. And so that's exactly what happens, right? So now we're only putting in the elongation map into the model, and the shape just flattens, it goes completely the wrong way. And there's essentially no residual, okay? And so, okay, so far so good. So far we think the cell rearrangements are an active driver of the shape change. They're not capturing everything, but we also now know that the cell elongations that we see are a passive back response to the active cell rearrangements, right? So there's still the isotropic channel, right? There's still the area changes. So we can put those in on their own and see what happens. And they do something funny. They sort of maintain this total flatness along the DV boundary that we wanted, but they have this funny like inversion of curvature around the tip because of the DV boundary itself. Here, so on their own, this is bad. But interestingly, in the residuals, you again get a component that should contribute to the anisotropic part. And so the idea then becomes, all right, what's likely going on is that there's a small amount of active area changes and a large amount of active T1 rearrangement, changes. And that you can balance them by balancing the residuals, you can figure out the relative ratio and put them together. So this is just showing you that the same band as before from just the rearrangements. And so yeah, now we want to put these together in a way that is sort of informed by the residuals. Wait. No, what happened? Yeah, and so yeah, so this is putting everything together now. This is our best combination, based on what is active and what is passive and what are the residuals. So this is active rearrangements and some partitioning of the area changes into active and passive, based on the residual matching. And then you could then compare the shapes, and now you pick up some of the negative downwelling, and this is much flatter. It's still not quite as flat as the real system, but it's much flatter than just the rearrangements. And there are still some overall residuals, but they're the things that are driving the elongation changes and the residual part of the isotropic changes. And so this is great, right? So we have one fitting parameter, right, where we fit I didn't show the sweep, but we did all this with the sort of fitting the bending modulus. And we're qualitatively reproducing the shape change, even with these highly symmetric pattern and only these net changes in the rearrangements and everything else. Okay? Questions so far? Okay. Okay. But so this, this is all well and good for the wild type, but maybe we did have one fitting parameter, maybe if the pattern gets changed somehow, we look at something else, maybe it's just not going to work. Maybe this is just a two-pat story just that happens to work in just this case. Okay. And so what if the patterns are screwed up? Right. So that's an important question. Like many times in biology, you think you show something, but then you really want to be able to show that your idea still works, if you have a perturbation. And so on the top, right, this is what happens in the wild type, right? So you have this, here's the wing disc, and it rotates. I didn't show you this before. There's these folds sort of up tissue that unfold during the process, and their unfolding kind of rotates rotates the tissue upwards in the way it's shown here. But the metric shape also changes considerably. So this up to 4, right, is a version, and this is the it just gets longer and closer together at later times. And so there's so Natalie and Suzanne, sort of had discovered this funny mutant in myosin 6 for a project where they were looking at sort of mechanical signaling in the stages prior to this, earlier times, where the first sort of evidence of that circular pattern begins to appear. I didn't even mention that, but yeah, there's some earlier work showed that there was this sort of circular pattern in cell behaviors, even earlier times. Yeah, so they discovered this myosin 6 mutant where essentially it doesn't destroy the rearrangements. You still see rearrangements if you look at it, but it destroys the pattern, right? So you no longer have a net set of rearrangements. And so then if you just qualitatively, what happens is, again, you still have the rotation, the folds still unfold, the whole tissue still rotates, but the shape almost doesn't change at all, right? The shape of the wing disc part. But you notice that it gets really, really flat and even curves in a little bit at the tip. So that's a shape change, but the rest of it doesn't do much. Okay, and so we can play the same game, right? We can take the myosin 6 mutant, we can classify what happens to the shape, go to the actual data, we can look at the cell behaviors, right? And in the context of the mechanical network, and see how much are the rearrangements in the topology, how much are the sort of the cell elongating or area changing. And then you can look at if you only put in the rearrangements that are there. And so notice, so I'm showing here the comparison between the wild type on the left and the myosin 6 on the right in each of these. So the elongations are pretty similar. The rearrangements go way down. And the cell area changes are similar, except in the DV boundary where something really weird happens we don't understand. So anyway, you only put in rearrangements, then you, much like in the wild type case, with rearrangements alone, you do already a great job at reproducing the shape across the DV boundary, but you do less well along the DV boundary. And then you put together the ingredients with the residual strains in the same way, and in order to include the spontaneous strains coming from the isotropic deformations, and then suddenly you have a great match, right? So now you have, again, across DBB is well explained, and along DBB is well explained, and I would argue that the model is validated by the perturbation. Right? So you're capturing the same shape changes and identifying the active and passive layers, with this approach. And so the take-home messages from the sort of today, the mechanical network part, and sort of what else can we do with this, is just to remind you of the cephalic furrow, right? The first biological example we talked about today is a mechanical sink, so that more spontaneous strains can happen at the same time in evolution. Right? You can do development faster, you can initiate more spontaneous strains faster. Actively driven rearrangements, in the cell network, can create effective spontaneous strains. It's theoretically possible. And that the wing disk conversion is driven by a pattern of exactly these kinds of rearrangements, in a system with a topological defect on top of it, right? So there's the topology of the network and then the topology of the topological defect in the coarse grained strain field, right? So there's this sort of multiple level interplay between geometry and topology going on. And I never mentioned this. And actually for this audience, it's not so important because, it's a very broad audience, but in the biophysics tissue mechanics field right now, tissue fluidization is a big deal, and it's way too big a deal, I would argue, philosophically speaking. My opinion is that it is important, but it's overblown, and that what I just showed you is the creation of complex shapes during development with zero tissue fluidization required. And many people will tell you, oh, if you see cell rearrangements at all, that means your tissue is fluid, right? That's nonsense, right? That is not true. And I think people know that that's not true, but they're just being, I think, too lax about it and not thinking deeply enough. When they say it, it's just easy, right? It's easy to communicate that to biologists. It's, fair enough. But I think this is an example where you have a system that's sort of deep in the solid regime of the tissue of the vertex model. It's a solid, it behaves as a solid tissue at all times, and yet it has this sort of, reshaping that occurs, mediated by presumably actively driven T1 events. that creates a complex shape, right? If a tissue were always fluid to create complex shapes, then the complex shapes would just melt, right? Like it doesn't work. So again, this is a little like a sort of advertising slash pedagogical slash philosophical slash, but again, for you guys, it's less important. So yeah, and then as far as other systems that we're thinking about with this, so the Hydra is a really cool system that has the sort of pneumatic order very clearly because they have these sort of supracellular bands of actin fibers that make topological defects. It's very interesting. It's very hot right now, actually. And so we have, we're thinking a little bit about Hydra. We're going to start to think about jellyfish, especially there's some observations that the way that jellyfish regenerate is strongly determined by the topology of what is cut off versus what is left, and understanding how the regeneration sort of proceeds relative to that topology, both of the mechanical network topology and the coarse-grained topological defect topology in both cases. This is, I think, a very interesting question. We're just starting to work on that. And then anisotropic growth of the fin, which is a multi-layered tissue, and the formation of the optic cup, which I think that's the story among these that's closest to being done, and is sort of related in the sense that the cool thing about the optic cup is, maybe just get a quick diagram. I know it's not here on the movie, but I'll just describe it in words. Is that the optic cup is like a big sphere, and then the apical surfaces are like right up against each other. So it's like one apical, like you have tissue, you have cells here and cells here, and then like you go around the bend. But the two apical surfaces, it's not like a lumen with a big ball of fluid inside. It's like the two apical surfaces are like two flat pancakes that are right on each other. And then this shape, to make room for the lens, it sort of has this big invagination occurs, and the apical surface even slides back further like this. But it's a double, again, it's this double apical surface. So like the mechanics of the apical surface, they're coupled potentially to these two apical surfaces together. And so you get, again, this sort of mechanical network, actually two interacting mechanical networks and the overall shape change effects going on. It's a really cool system. And so we're working on it, and that's the one of these that's kind of closest to having a definitive answer on, which I'm trying to understand how it works. And then just a little advertisement for, so for like the coarse band shape changing sort of spring lattice model that we use, we implemented in this software platform together with Frank and Frank Ulicker and Ivo Spalterini called TopoSpam. It's available at topospam.org. There's gonna be a methods paper on this. We're writing it up now. But the idea is that like it includes this, the software package to run these sort of coarse-grained tissue shape change models together with polar active gels, polar active fluids, and 3D and quasi-3D vertex models. So it's like a one spot shopping spot. It's not shopping, it's free to download, but one spot source for many different scales of topologically sensitive mechanical biology models, like shape sensitive, topology sensitive biology models. So for like polar active fluids and gels for sort of cell cortex and intracellular things, 3D quasi-3D vertex models for like interacting cells and sort of up to some number of cells, and then coarse graining over cells with whole tissues, right, for the spring models that I showed you before. And then same advertisement I've shown every day, just one more time for good measure. If you are thinking about trying to find a postdoctoral landing spot, then do think of us. The ELBA postdoctoral fellows program it's a really good one. And again, for faculty, the visitors program is also really fun. Really fun to have people around. And now, because it's the last day, I should thank lots and lots of people. So this is sort of focused, like the focus here is on the ones I talked about today, but also many collaborating labs that I talked about in the last 2 days, my own group. So today I showed work from Abhijit, who's here, Alicia, who's not pictured, Carlos, who's there. the theory of the opposing apical surfaces in the object cup I just talked about is being done by Lior. There. Some of the network stuff, so Felix and Chinmayi, so Chinmayi is there, Felix is pictured. Did the intertwined and snarled networks. Sherbalch was a previous postdoc with us that was doing the beta skeleton stuff. So Pavel Tomanchak in his lab with the sphenic furrow, Suzanne Eaton, Natalie Dye, Frank Buliker, Mark Popovich, all collaborators on the flywing tissue stuff. Topospan with Elo and Frank again. For the network stuff, mostly with Marino and Anke Panaton. Jellyfish with Sengali and Shimonovich. The brief slides on the diatoms from the first day, I think, that was with Ms. Kroger, and also Shavelch, and many more. And yeah, so I thank you guys for coming and listening these 3 days, and I'm happy to take some questions. We have a few minutes left on the session. So, thanks.
Thank you.